Okay, good evening, everybody. A very warm welcome to each one of you. Thank you for joining us today. So, as we know, Deborah and Rise movement was initiated by women during the COVID period uh, to to bring together people from different walks of life who wanted to serve during the crisis period. So at DRM, we have different platforms. We have the Life Connect, Social Connect, and the Prayer Connect. So through Life Connect, we attempt to answer various questions of life that people have and have raised with us at different points in time. Questions like, is there a God? What is the purpose of life? Health? How do we sustain health? So on and so forth. And we've brought through in this platform, we've had many eminent speakers who've come and shared with us their knowledge. And today we have amongst us Dr. Jonathan, who will be helping us to understand how biological information points to design. Okay, Dr. Jonathan is an assistant professor at the Salter College, Boston, and he's a fellow of the and he's also a member of the Discovery Institute there. He's the founder of talkaboutdoubts.com, a ministry that attempts to answer questions but regarding the gospel. So today, thank you, Dr. Jonathan, for making time for us. And it's a privilege for us to have you here with us. Over to you, Dr. Jonathan.
All right, hopefully you can see my screen. Well, thank you so much for having me again to talk about how biological information points to design, which is the topic of our discussion today. Just a little bit of background for those of you who haven't been in the previous sessions that I've done for this group or aren't familiar with my work. So I'm Jonathan McClatchy. I am, as Dr. Christie said, I am an assistant professor at Sattler College in Boston, Massachusetts, and I teach various courses relating to biology, including freshman biology. I teach sophomore genetics and genomics. I also teach bioethics. I teach microbiology. So I have a PhD in biology from Newcastle University in the UK. You can find my website at jonathanmcclatchy.com. That features many of my essays relating to various aspects of the defense of Christianity in particular, and theism more broadly, including articles relating to design in the life sciences too. So if you want to check some of my articles and videos out, then you can find a lot of that stuff at jonathanmcclatchy.com. And if you want to contact me later, my email address is there on the slide, jonathan@jonathanmcclatchy.com. I also I'm the founder and director of talkaboutdoubts.com, which is an online ministry that specializes in offering free private mentoring and discipleship to Christians who struggle with doubts in relation to the veracity of the Christian faith. And so if you are going through a season of doubt and you have questions and concerns about whether or not Christianity is in fact true, then I would encourage you to go to the website talkaboutdoubts.com and get in contact with us there. Your email will will be distributed to one of our scholars in a relevant subject, and that individual will get in contact with you to talk to you in private about your doubts and concerns. And so we have a diverse pool of thinkers from various disciplines, including physics, biology, biochemistry, New Testament studies, Old Testament studies, Hebrew, biblical archaeology, psychology, therapy. We have pastors on our team. So yeah, even if you know someone that this resource should be of benefit to, then please feel free to pass on this information to them. We also talk to a lot of parents about their kids and try to help as we can with for parents whose kids have walked away from the faith. We also talk to people in full-time ministry positions as well. So feel free to reach out to us if you think that could be of benefit to you. So let me then, without further ado, launch into today's topic, which is how biological information points to design. Now, before we can assess whether a particular feature of the world constitutes evidence for a hypothesis, we really need to determine what evidence is. What do we mean when we say that X is evidence for Y? What does that actually imply? Well, imagine that we have a court scene and the expert witness comes forward, or the forensic scientist or the detective, and he lays out the murder weapon which was used in a particular murder of which the defendant is charged. And he observes that the fingerprints of the accused actually are on the handle of the murder weapon. Now, does that prove that the defendant is guilty of committing the murder? Well, no, not necessarily. There could, in principle, be other explanations for why his fingerprints ended up being on the murder weapon. But it is evidence that tends to confirm or tends to support the hypothesis that he is, in fact, guilty of committing the crime. So the presence of the accused's fingerprints on the murder weapon is evidence for the guilt hypothesis over the non-guilt hypothesis, because it is more probable given guilt than given non-guilt. It's more likely, if he's guilty, that his fingerprints would be on the murder weapon than it would be on the falsity of that hypothesis. Could you guys mute perhaps while I'm giving the presentation, because there's a lot of feedback in the environment. Thanks. So anyway, the presence of the accused's fingerprints on the murder weapon is evidence that they confirm the hypothesis that the defendant is guilty, because that hypothesis renders it more probable that the accused's fingerprints would be on the murder weapon than it would have been otherwise, and that in turn raises the likelihood of the guilt hypothesis. So that's really what we mean when we talk about evidence. When I say that X is evidence for Y, I don't necessarily mean that X entails Y. What I mean is that X raises the probability of Y relative to what it would have been otherwise. Now, what sort of evidence are we dealing with when we look at biology and the world of the cell? Well, I'm here today to talk about the biological information, and the most obvious place to begin when it comes to biological information is the information storage medium that we call DNA, or deoxyribonucleic acid, which you see pictured here. And the DNA is a language, has a language convention called the genetic code, where triplets of nucleotide or triplets of letters that are known as codons basically specify amino acids that form proteins, and by virtue of the side chains of the amino acids in a chain of amino acids, that will determine how a protein will collapse into a three-dimensional structure that can fulfill its job in the cell. But that's determined by the sequence of DNA, and DNA is a four-letter code. It's comprised we represent four chemical subunits with the letters A, C, T, and G: adenine, thymine, guanine, and cytosine. And the sequential arrangement of those alphabetic characters that represent those chemical subunits is functionally meaningful. And so we're dealing with digitally encoded information that runs along the spine or the sugar phosphate backbone of the DNA molecule. And information content, I would argue, is something which is habitually associated with conscious activity, with rational, conscious deliberation, intelligent design, agency. Whenever we find information, particularly in digital form, in any realm of experience, whether we're talking about hieroglyphic inscription or a computer programming script or whatever it happens to be, habitually is associated with conscious deliberation. And therefore, the hypothesis that a mind was involved in the origins and development of life on our planet renders it not particularly surprising that we'd find information content in the cell, because that's something that we typically associate with consciousness. However, on the falsity of that hypothesis, the idea that there is no mind involved in the origins and evolution of life on our planet, then that renders the observation that we find this biologically meaningful information content in the cell wildly surprising, wildly improbable. And so you have this top-heavy likelihood ratio where the evidence in question, namely the biological information, is far less surprising given the hypothesis of design than it would be on the falsehood of that hypothesis. And therefore, it tends to confirm the hypothesis of design. It raises the likelihood of the design hypothesis relative to what it would have been otherwise. So as I said, the information DNA directs the construction of proteins in the cell. Here's a short animation that shows the flow of information from DNA to messenger RNA to proteins, which is known as the central dogma of molecular biology. And this short animation reveals the complexity and engineering prowess of the information processing apparatus that we find in the cell.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
Okay, so that was that animation that you just saw depicts the direction of information flow, which is known as the central dogma of molecular biology. So the central dogma of molecular biology was first advanced by Francis Crick, who was the co-discoverer with James Watson of the double helical structure of the DNA molecule, and the central dogma describes the flow of information from DNA to messenger RNA to protein. So DNA makes RNA, makes protein, makes us. So this term, as I said, was coined by Francis Crick in 1958, and it indicates the basic flow of information from DNA to RNA to protein. And the DNA is made up of nucleotides, which nucleotide bases, which in turn determines the sequential arrangement of the amino acids that comprise a protein and will determine how the protein will fold into a three-dimensional structure. So as I said, the sequential arrangement of amino acids determines how a protein folds. And so you can see you've got the primary structure, which is a sequence of the amino acids itself, and then that determines how the protein folds into secondary units of structure known as alpha helices and beta sheets, and then these come together to form the tertiary level of structure, and then many proteins that are used in the cell are multi-domain proteins, which is the protein's quaternary structure, where you have these multiple folds that come together to form the quaternary structure of a protein. So how evolvable are protein folds by evolution, by natural selection and chance variation? Well, there are 20 different amino acids are used in cellular proteins. And that means that for a modestly length protein of 150 amino acids, there are 20 raised to the 150th power of possible combinations. And so a question that we can ask is, how prevalent are functional protein folds in combinatorial space? Out of that 20 to the 150th power of possible combinations, we know that many of those are functional, many of those are non-functional. And so the question that we want to ask empirically is, what is the ratio of those stable and functional protein structures within that sea of combinatorial possibilities? I mean, to put that number in context, there's only 10 to the 80 elementary particles in the known universe. So 20 to the 150th power is a huge combinatorial space. Now, we know that in the English language, words communicate meaning, because they, by virtue of the fact that meaningful words, sentences, and paragraphs are astronomically rare relative to the space of gibberish, right? There's a lot more ways of writing nonsense than there are ways of writing meaningful sentences and paragraphs and so forth. And the same thing seems to be true in biology as well. And if that's the case, then, and this is a question that can be investigated empirically, then it would seem to me to be problematic that by an unguided stochastic process that is devoid of any form of foresight or mind, that you could find those functional sequences by a chance type process in order for natural selection to work on that, to preserve those advantages that are attained by those stable and functional protein structures. Well, there's been papers published on this subject. For example, this is a paper published in Nature titled "Robustness Epistasis Link Shapes the Fitness Landscape of a Randomly Drifting Protein." And of course, they conducted a site-directed mutagenesis experiment to assess the sensitivity of the protein to changes or substitutions in the amino acids that comprise that protein. And what they discovered is that after only a few random mutations under weak selection, around a third of subsequent changes to a protein completely disable it. After several more mutations accumulate, the protein is inactivated by slightly under 2/3 of subsequent changes. And after random alteration of less than 10% of the protein's initial sequence, it becomes permanently non-functional. So the fitness approach is 0. Now, the corresponding rarity can be evaluated by back-solving or working backwards. So the number of sequences that differ from an optimal one by a given number of amino acids increases almost exponentially with the number, such that a random search would find a barely functional sequence long before an optimized one. And so estimating the upper limit for the probability of a successful trial must focus on that neighborhood of sequence space. So let's do the math then. So in the study, after 5 to 10 mutations, roughly 2 in 3 mutations inactivate a protein. Therefore, 1 in 3 amino acids at each position on average would correspond to a functional sequence. So that rarity would be less than 1/3 to the power of the sequence length. So for example, if you have an amino acid sequence that's 150 amino acids long, then the rarity is going to be something like 1/3 raised to the 150th power. That estimate closely matches the result of Doug Axe's 2004 beta-lactamase experiment, where he also conducted a site-directed mutagenesis experiment, and he determined that the rarity was something like 1 in 10 to 77 sequences correspond to a functional fold or domain within the protein. And the actual rarity actually is much more extreme than that, since almost no sequences are functional after 10% of a protein randomly changes. And this was Doug Axe's paper that he published in the Journal of Molecular Biology, based upon similar principles to the paper I just alluded to from the journal Nature. So if the protein structures are astronomically rare within that sea of combinatorial possibilities of ways of arranging amino acids, right? There's 20 different amino acids used in life. And so, there's 150th, the 20th, 150th power of possible ways of arranging a 150 amino acid long residue. And we find that the vast majority of those sequences are functionless. They aren't able to perform chemistry, aren't able to form a stable protein structure. Then how by a stochastic unguided process would you be able to find those sequences, given that you're much more likely to find gibberish? And natural selection, of course, while it ensures the survival of the fittest, it cannot drive the arrival of the fittest. For that, you need an unguided blind search through sequence space. This presents us with a probabilistic resource problem. So there are approximately 10 raised to the 80 elementary particles in the observable universe. And since the Big Bang, roughly 10 raised to 16 seconds have elapsed. And the time it takes light to traverse the smallest distance, or Planck length, of 10 to the minus 33 centimeters is 10 raised to the minus 43 seconds. So by multiplying together those three relevant factors, we can determine that the maximum number of events that could have taken place in the observable universe since its origin is 10 raised to 139th power. This is what's sometimes called the universal probability bound. Anything that exceeds that probability, anything that's less probable than 1 in 10 raised to the 139th power, is beyond what can be afforded by the probabilistic resources of the entire universe. That universal probability bound is swamped very, very quickly, especially when we're dealing with complex molecular machines requiring multiple proteins to work in tandem. So in conclusion, then, locating the functional sequences within the vast sea of combinatorial possibilities by a random search is like finding a tiny needle in an enormous haystack within a very limited time. The problem, though, extends deeper than that, as I just mentioned. When molecular machines require multiple cooperating proteins in order to perform their function, the probabilities multiply exponentially. So here we don't just need any protein that performs some sort of chemistry, but you need very specifically crafted proteins that have to work together in unison to achieve a higher level objective. In the case of DNA replication that I pictured on the slide, you need proteins that can work together to achieve the higher level purpose of replicating or duplicating DNA. And if any of those components is missing, such as the DNA polymerase or the sliding clamp or the clamp loader or the primase enzyme or the single-stranded binding proteins that prevent the DNA double helix from reannealing during copying, or the topoisomerase enzyme that alleviates supercoiling induced by the torsional stress, and so forth. If any of those protein components, and many others like helicase that breaks the hydrogen bonds ahead of the polymerase, if any of those were missing, you wouldn't have DNA replication that worked half as well as it used to, or quarter as well as it used to, but it would be broken. And so how do you get, by an unguided process that doesn't have foresight, all of the relevant proteins that are necessary to be very specifically crafted to work together in unison to achieve a higher level objective? It strains credulity, I think, that this could be plausibly accounted for by a stochastic, unguided natural process, but it's not particularly surprising we'd find such features of the world on the hypothesis that a mind was involved in life's origins and evolution on Earth. And so we have a top-heavy likelihood ratio, and therefore this sort of evidence tends to confirm the hypothesis of design relative to the hypothesis of non-design. As I mentioned before, there's a language convention that's used in life. This is known as the genetic code. The words in DNA language are spelled with 3 letters, and these 3-letter words are known as codons, and there's 64 different codons, and each of those codons specifies an amino acid. And there's a redundancy in the genetic code because there's 64 different codons, only 20 amino acids, so you end up with a situation where amino acids can be specified with multiple codons. And there's 3 stop codons which signal to stop translation by the ribosome. And these stop codons are UAA, UAG, and UGA. There's also a start codon which is UGA, which is AUG rather. The stop codon is AUG, which codes for methionine, which is almost the first amino acid that's used in biological proteins. Okay, so that is the genetic code. Now, the genetic code appears to be very highly optimized for minimization of errors that could be incorporated by substitution mutations. And there's actually multiple levels of fine-tuning of the genetic code. The genetic code found in nature actually is exquisitely tuned to protect the cell from the detrimental effects of substitution mutations. In fact, the system is so brilliantly set up that codons differing by only a single base either end up specifying the same amino acid or an amino acid that's a member of a related chemical group. So in other words, the structure of the genetic code is set up to mitigate the effects of errors that might be incorporated during translation. Another astonishing feature of the genetic code is that the sequences of stop codons overlap with those of the codons specifying the most abundant amino acids. So an example of that, I'll just show the table again. An example of that would be, so glutamine is the most commonly used amino acid, and you can see that the codon is GAA and GAG. You see that these actually differ by only one letter from those those codons that are stop codons. Another popular codon that's used is that specifying glycine, and you can see again these differ by only one letter from the stop codons, and that's deliberate. That seems to be intentional, because it means that the genetic code is set up in such a way to dampen the impact of frame shift mutations. Frame shift mutation is basically where you have an indel, which is an insertion or deletion of a number of nucleotides that's non-divisible by 3, and it basically shifts the whole reading frame, which could wreak havoc because it results in the production and accumulation of misfolded proteins. And the earlier on in the sequence that the indel occurs, the greater the alteration of the protein's amino acid sequence. So this feature of the genetic code, this level of fine-tuning, essentially ensures that when you get an indel insertion or deletion that's not divisible by 3, then it results in the formation of stop codons that shuts down translation, prevents the protein from being translated. And so that's also a design feature, I think, of the genetic code. So this paper argued that in the conventional genetic code, translation of frameshift errors halted faster in errors than in 99.3% of alternative codes. Related to that is the ability, also reported by the same paper, of the universal code to allow arbitrary sequences of nucleotides within coding sequences much better than the vast majority of other possible codes. It's not just me saying this, there's a whole wealth of literature on the fine-tuning of the genetic code. Here's just one example from the Journal of Biosystems titled Optimality in Standard Genetic Code is Robust with Respect to Comparison Code Sets. Now, how might you evolve a genetic code? How might you optimize a genetic code over time if you're using a kind of a trial and error process? Well, changes in codon assignments, that's the mapping between codons and amino acids that they specify, would actually be catastrophic to the cell, because such a mutation will ultimately lead to changes to the amino acid sequence in every protein produced by the cell. And that means that one cannot have a significantly evolving genetic code over time, because you're just going to, you're having to change every polypeptide made by the cell if you change those codon to amino acid assignments. And so that makes this enormously implausible to account for by a trial and error type of process. Now, some have tried to argue around that by positing that the lesser used codons could be redesignated to a different but related amino acid, that's allowing the genetic code to become optimized. But there are, I think, significant difficulties with that suggestion. For one thing, it seems quite unlikely to me that by virtue of replacing some of the lesser used amino acid assignments with a related amino acid, that one could attain the level of optimization that we find in the conventional code. Furthermore, the question is naturally raised as to what selective utility would be exhibited by the new amino acids. In fact, they would have no utility until incorporated into proteins, but that won't happen until they're incorporated into the genetic code. And there's also, of course, the added problem of the potential for codon mapping ambiguity. If for example, 80% of the time a particular codon specifies one amino acid and 20% of the time specifies another, that mapping ambiguity also would lead to cellular chaos. So it's a really significant challenge, I think, to a naturalistic account of life's origins, and actually points to design, because this sort of optimization of the genetic code is something that's not particularly surprising on the supposition that a mind was involved, but it's wildly surprising on the falsity of that hypothesis. Now, let me talk a little bit about how chromosomal DNA is packaged, how it's organized in the cell. So So DNA in the nucleus is spooled around these histone octamers. Histone octamer consists of 2 copies of each of the 4 core histone proteins. These are known as H2A, H2B, H3, and H4. And the octamer assembles when a tetramer, which contains 2 copies of both H3 and H4, complexes with 2 dimers of H2A, H2B. So these are histone octamers that the DNA spools around, and each core histone protein has a tail that extends away from the nucleosome core between the columns of DNA. And these tails are stretches of amino acids of up to 25 amino acids in length, and they don't adopt a defined structure. And the histone tails interact with adjacent nucleosomes to condense the chromatin structure further, but their precise role in chromatin compaction remains an area of active study. So chromatin is basically just the structure of the DNA in the cell, and the chromatin is the structure of the proteins and the DNA together that make up chromatin. And chromatin can be either loose or it can be condensed depending on whether the genes in that particular region are being expressed in that cell type or not. So chromatin is organized into filaments, as you can see. This is the nucleosome, which the DNA is spooled around these histone molecules, and you can see that these form this very condensed structure. This is known as heterochromatin when it's highly condensed like that, whereas if it's more loose, it's known as euchromatin. And euchromatin tends to be where the genes get expressed in a cell type, and heterochromatin is where they are shut down when they're not being expressed. Okay, so histone H1 binds to the linker DNA that connects successive nucleosomes, and that facilitates the compaction of the chromatin. So histone H1 is known as a linker histone. And the tails of the core histone proteins that protrude from each nucleosome are also important in the formation of the 30 nanometer fiber, which we saw here, this very thick, this very highly condensed fiber. But the precise way in which they stabilize the fiber remains an open question at this point. Now, so chromatin is organized into filaments. So in the cell, the 30 nanometer fiber is further organized to become part of the compact forms of chromosomes that are seen in electron micrographs, as we see here. But we know little about the precise nature of how chromatin is further compacted to form these structures. DNA packing occurs on several levels in chromosomes. So you've got the lowest level of compaction, which is just the short region. This is a short region of the DNA double helix, which is 2 nanometers in diameter. Then you've got the beads on a string form of chromatin, which is 11 nanometers in diameter. You've got the chromatin fiber of packed nucleosomes, which is 30 nanometers in diameter, right? The 30 nanometer fiber. And then you've got the chromatin fiber, it gets folded into loops. And then you've got the entire mitotic chromosome, which is 1400 nanometers in diameter. So very, very highly compacted, very condensed, and this the DNA gets organized into this very condensed structure in preparation for mitosis, preparation for cell division. So the net result is that each DNA molecule has been packaged into a mitotic chromosome that is a 10,000-fold shorter than its fully extended length. There's variation in chromatin structure within the cell. So some of the chromatin is very highly condensed, and some of it's is more uncondensed. So euchromatin is the uncondensed form. Heterochromatin, which is darker here in the image, is the very condensed form. And if a particular sequence is not being expressed in that cell type, then it will be among the heterochromatin. If it is being expressed, it will be among the euchromatin. And then there's also the nucleolus in the middle of the nucleus here, where the RNA, the ribosomal RNA is produced. So these histones can be covalently modified by chemical modifications that determine whether the compaction of the chromatin is going to become more or less. So histone proteins that package DNA into chromatin are subject to this variety of post-translational modifications. Basically these modifications add chemical groups or small proteins to histone side chains, and they serve as important signals in regulating and controlling virtually all processes that involve interactions with chromatin. And these modifications occur both in the histone tails and in the globular core of the histone proteins. And once modified, these sites are thought to recruit specific proteins to the chromatin, which then affect chromatin structure and function. And the covalent modifications also constitute chemical changes that can alter chromatin structure directly. For example, acetylation removes the positive charge from the lysine side chains and thereby alters the interactions with the negatively charged DNA. Histone acetylation is associated with active chromatin. So virtually all regions of eukaryotic chromosomes contain histones with acetylated lysine residues. So modifying the lysine amino acid with an acetyl group. But the relative levels of acetylation vary quite a bit. So activated transcribed regions in euchromatin, the loose form of chromatin, contain highly acetylated histones, whereas the heterochromatin, the more condensed form of chromatin, which contains few transcribed genes, has very low levels of acetylation. And the primary targets of acetylation are histones H3 and H4, and these contain numerous lysine amino acids that can become acetylated. And there are enzymes that are known as histone acetyltransferases that can attach these acetyl groups to the lysine side chains, while the histone deacetylases remove the acetyl groups. So here's just some classes of histone modifying enzymes in yeast and humans. And the histone tail acetylation, so attaching these acetyl groups to the histone tail, allows recruitment of proteins that act on chromatin. So when the histone gets acetylated, when these acetyl groups get attached to the histone tail, that generates sites for the binding of proteins that contain domains known as bromodomains, and these domains bind to particular acetyl lysines, and the bromodomain protein can then recruit other proteins such as the nucleosome remodeling complexes or other enzymes that act on the DNA. And the effect of acetylation can also derive from the resulting change in the electrostatic charge, because unmodified lysines are positively charged, whereas the acetyl lysine is electrostatically neutral. So the histone tail methylation, so we just covered acetylation. So histone tail methylation, so modifying the tail by attaching a methyl group, recruits the proteins that initiate heterochromatin formation, so the more compact or condensed form of chromatin. So that so here you can see the attaching of the methyl groups, and then the chromodomain protein, and then the recruited protein or protein complex, and that results in the formation of the more condensed form of chromatin known as heterochromatin for gene silencing. You've also got histone phosphorylation, adding phosphate groups to histone molecules. So histone tails can be phosphorylated at either serine or threonine amino acid residues. And the phosphate groups are added by kinases and removed by phosphatases. And histone phosphorylation also plays a variety of roles, such as, for example, phosphorylation of histone H3 at serine 10 alone allows the transcription of genes required for cell growth. Chromosome condensation and mitosis is tightly correlated with phosphorylation of histone H3 at both serines 10 and 28. Phosphorylated residues can be recognized by specialized protein domains, although the precise mechanism by which phosphorylation of histone exerts an effect remains to be elucidated. Another modification is attachment of ubiquitin peptides to histone, to the histones. So the covalent attachment of ubiquitin to lysine side chains is the largest histone modifications, since ubiquitin is a 76 amino acid protein. And ubiquitin is attached to histones in a series of enzymatic steps that can be removed, and it can be removed by a deubiquitylating enzyme, or DUBS for short. And histones can be similarly modified by the covalent attachment of SUMO, or small ubiquitin-like modifier, which is similar in structure to ubiquitin. And so when histones get ubiquitylated, it plays roles in regulating different steps of transcription as well as DNA repair. And modification of one site can often influence the modification of a second site, as we see here. So post-translational modification of one histone protein can trigger a cascade of events that leads to further chromatin modifications, either in another histone protein or in the DNA itself. So, for example, phosphorylation of serine 10 promotes the acetylation of H3 lysine 14, which is associated with active chromatin. Acetylation of H3 lysine 14 then inhibits methylation of lysine 9, which is a docking signal for proteins associated with heterochromatin formation. Now, chromatin remodeling allows access to specific sites on the DNA. So the binding sites for specific proteins in DNA can be inaccessible to DNA binding proteins owing to because of the association of the region with the nucleosome. So ATP-dependent chromatin remodeling results in the movement of the nucleosome along the DNA, which then exposes the binding site, which allows the DNA binding proteins to bind and exert their effects. So there are different ways in which nucleosomes might be repositioned along the DNA. There's a number of models that have been suggested for how nucleosome remodeling enzymes reposition nucleosomes. So in this first one at the top of the slide, we've got a propagation of local DNA distortion around a histone octamer can lead to sliding of the octamer along the DNA. And the second model, this is a global conformational change in the DNA around the histone octamer could result in or trigger the repositioning of the nucleosome on the DNA. Okay, so once it's been established, heterochromatin, the more compact form of chromatin, can spread because these histone modifications attract a set of heterochromatin-specific proteins, including histone modification enzymes, which then create the same histone tail modifications on adjacent nucleosomes. And these modifications, in turn, recruit more of the heterochromatin-specific proteins, which causes a wave of condensed chromatin to propagate along the chromosome. And the heterochromatin will continue to spread until it encounters a barrier DNA sequence that stops the propagation. And basically, the barrier sequence prevents crosstalk between adjacent chromatin domains. So there's these DNA sequences or barrier sequences that result in this barrier between the chromatin domains and prevent crosstalk. And several of these have been identified through genetic engineering techniques, and they can add or delete DNA sequences to or from chromosomes. So, for example, in red blood cells, the beta globin locus in decondensed or activated chromatin is separated from an adjacent region of condensed or silenced chromatin by a sequence known as HS4 and so forth. And if you delete that sequence, it results in the beta globin locus being invaded by condensed chromatin, and the result of that is quite catastrophic because the globin genes are not well expressed, and individuals that carry that deletion actually suffer from severe anemia. So because nucleosome remodeling is typically required in precise locations on the DNA, the cell has to have a mechanism for recruiting nucleosome remodeling complexes to specific sites in the DNA where they're needed. And the most common way of recruiting these complexes is through association with proteins that bind to specific sequences in the DNA. And a wide variety of eukaryotic transcriptional regulators can recruit nucleosome remodeling complexes through direct non-covalent binding between proteins. And post-translational modifications in histone proteins can also recruit nucleosome remodeling enzymes. Okay, so this just shows the sliding of the DNA. This is an ATP-dependent chromatin remodeling complex that is driven by ATP hydrolysis that generates energy and allows you to remodel the chromatin to expose a particular region of DNA that needs to be expressed. And methyl group, so DNA methylation can recruit proteins involved in histone modification and vice versa. So methyl groups, which are shown with the blue triangles in this figure, can be added to both the DNA itself as well as to histone tails. So in this example, this is the DNA methyl binding domain, and methyl groups on the DNA recruit these methyl binding proteins, and that can in turn recruit histone deacetylase, which is shown in the orange here, which modifies the chromatin by removing the acetyl groups, which are shown with these pink squares, from the histone tails, and it thus initiates silencing. Methyl binding proteins that have histone methyl transferase activity, which is shown with the green, can bind to sites of the DNA, can bind to sites of DNA methylation and methylate the histones, which are shown with the blue triangle here. And that is a signal for chromatin silencing. The methylation, so in this one, the methylated histone tails can bind the chromodomain containing proteins, which are shown in green, which in turn results in recruitment of DNA methyltransferases that methylate adjacent DNA. And then finally, the nucleosome remodeling, in this one, the nucleosome remodeling complexes, which are shown in this dark blue protein, can bind specifically to regions of methylated DNA, and using subunits that specifically recognize methylated cytosine. So it's a very, very complex process that is basically, this is information content beyond the realm of the DNA molecule in the form of these covalent chemical modifications of histone tails and so forth. So what are the implications then for intelligent design? Well, whenever we discover information-rich or language systems, the search space is far, far too vast to be sampled in the time available in the history of life. To the contrary, the molecular processes discussed in the foregoing show every evidence of intentionality and deliberate design and planning. So, for example, histone H3 contains 19 lysines known to be methylated. Each can be unmethylated, monomethylated, dimethylated, or trimethylated. And if modifications are independent, that allows a potential of 419 or 280 billion different lysine methylation patterns, which is far more than the maximum number of histones in a human genome. And that doesn't even include lysine acetylation, which is known for histone H3 at 9 residues. It doesn't include arginine methylation, which is known at 3 residues in histone H3. It doesn't include phosphorylation of threonine, serine, and tyrosine, which is known at 8 residues for histone H3. And that's not even to mention modifications of other histones besides histone H3. So there's a huge combinatorial space for these chemical modifications of histones. It also exhibits irreducible complexity, which we've talked about, I think, before in these sessions. So there's not much use in having a mechanism of writing these marks onto nucleosomes unless there's simultaneously a mechanism for reading them. The histone code system has to furthermore evolve simultaneously with insulator or barrier sequences to prevent unwanted or confusing crosstalk between adjacent chromatin domains. There's also the feature of causal circularity, which is basically a fancy way of saying a chicken and egg paradox. So the initiation of gene expression depends upon specific modifications to histone tails. But these histone modifications are themselves a result of gene expression decisions, which are themselves dependent upon the histone code. So, so it's a question of which came first, the chicken or the egg. That's a classic causal circularity problem. So, in summary, then we've seen that the information content in the cell, in particular in the realm of the DNA molecule and how that specifies the sequential arrangement of the amino acids that form proteins, as well as these epigenetic sources of information, these chemical modifications of histone tails that determine that influence and drive chromatin remodeling, is, I think, far better explained by design than it is by naturalistic, unguided stochastic processes, by virtue of the fact that the information-rich systems are far less surprising, given the hypothesis of design, than they are on the falsehood thereof. And so I'll conclude with that, and I'm happy to take any questions. Thanks for your attention.
Thank you, everyone. I'm just going to unmute everybody. So if anybody has any questions, you can just straight away ask Professor. I think there was a question by Mr. Krishnamurthy, I think. He had asked, not understood why the 3 should be multiplied. And he says maybe it's higher math.
Yeah. So that was in relation to the rarity of protein folds, I think, was it? I think he's referring to the allusion that I made to the paper published in Nature, Robustness Epistasis Link Shapes the Fitness Landscape of a Randomly Drifting Protein. So just to go through that again, so in that study, so after 5 to 10 mutations, there were roughly 2 in 3 mutations inactivate the protein. And so it follows then that 1 in 3 amino acids at each position on average would correspond to a functional sequence. Because if 2 in 3 mutations inactivate the protein, then 1 in 3 amino acids at each position on average corresponds to a functional sequence. And so the rarity is then less than a third to the power of the sequence length. And so that's, so if you're talking about, for example, 150 amino acid long sequence, then that's going to be a third raised to the 150th power. Does that make sense?
Could you define epigenetics in a simple layman's term?
So epigenetic information is simply information that's beyond the purview of the DNA molecule.
Sorry, I missed that. I'm driving, so could you just repeat it?
Yeah, it's information that's beyond the purview of the DNA molecule.
Beyond the purview. Yeah. So the question comes from a paper I read where they refer to being able to change the color coat of the hamsters by exposing them to a certain type of environment. Does that come under the definition of epigenetics?
I'm not sure what they did there. It might be that they used a gene from another organism.
They use the term there. They use the term there. They use the term epigenetics in the paper, and it was a paper to show that the exposure to environment affects gene expression.
Mm-hmm. Yeah, that would be an example of epigenetic information.
That would be an example of epigenetic?
Correct.
Right. So, okay.
Epigenetics is an umbrella term for any source of biological information that is not DNA.
No, but the color coat of the hamster, changing of the color coat of the hamster could not change unless the code was already available in the hamster's DNA.
Okay, well, if it's a DNA sequence and it's not epigenetic.
I said, but if the hamster's, the color of the hamster's coat changed, the expression should have been within the within the genetic material.
Okay, if that's
not extra, it is not an extra DNA expression, wouldn't it be?
Okay, then it's not epigenetic.
Okay. Just one last question. So in this whole genome and genome expression, if we are to boil it down to a simple thing where the layman could understand, it uses 4 nucleotides and maybe carbon, hydrogen, oxygen atoms in majority.
I'm sorry, say that again.
So in the whole thing of the genome and genome expression, the main what should I say, ingredients that go into make it is 4 nucleotides.
Right.
And if we say the atoms, it's carbon, hydrogen, and oxygen atoms in majority.
Yeah, exactly.
Right. Thank you.
Mm-hmm.
Thank you.
In all this, I don't know how to connect the intelligent design to this molecular, all those things. I'm just not able to understand that connection.
Yeah.
Jonathan, he's asking like, how would you connect that there is an intelligent designer through this explanation of what you gave? That is just a simpler way if you can tell them.
In regards to what?
No, in regards to this complexity, biological complexity, how everything points to an intelligent designer behind it all.
Sure. So as I said, I'm a Bayesian, so I think about evidence in terms of a likelihood ratio. When we're assessing how strong a piece of evidence is for a hypothesis, we want to know how likely is that evidence given the hypothesis, and how likely is that evidence given the falsity of the hypothesis. And when it comes to information content in the cell, whether it be DNA or otherwise, information content is something which is habitually associated in our everyday experience with conscious minds, with rational, intelligent deliberation. And so when we find information content in the cell, it is more likely that that information content would be there on the hypothesis of design that it would be on the falsehood. In other words, it's not particularly surprising that you'd find information content on the hypothesis that a mind was involved, but it's wildly surprising on the falsity of that hypothesis.
So
Yes.
you mean that, just for example, if there is a dictionary, there is an information. So you need a designer or a person, intelligent person, to make the dictionary. In the same way, in the cell, there are a lot of information, so signature in the cell. So there is a person who has signed it, who has done it. So without that, the information cannot come. That's what I think Jonathan is telling.
Correct.
Right?
Correct. I like Jonathan's example of a word. Like a word has meaning only if it is put in a particular sequence.
Right.
Yeah,
Exactly.
I like that
There's,
example, sir.
yeah,
So
there's
a
far
word has
more ways.
meaning only if it is, yeah, and that is a word is out of consciousness and intelligence. So similarly, DNA molecules, are in a sequence and expressed. That's how I understood it.
Any other questions?
Thank you, sir. Thank you, sir.
All right,
I also like the expression of, evolution explaining, what did you say? Does not explain arrival, and then before.
It explains the survival of the fittest, not the arrival of the fittest.
Yeah, I have to just recollect that. I'll check the recording. Thank you, sir. Lovely. I loved it. Thank you.
Thank you.
So thank you everyone for joining us today, and thank you, Dr. Jonathan, for sharing about what you shared today. Okay, there's another question just popped out. How can we disprove the self-arranging molecules theory by somebody called Joey?
Yeah, so if you want a detailed discussion of that, I recommend Signature in the Cell by Stephen Meyer, where he covers the self-organizational theories of Stuart Kaufman and others. But basically what we're dealing with in the cell is not regular repeating patterns that can be accounted for by law-like regularities. Rather, what we're dealing with is specified complex and functionally specific irregularity. So I do think that, there's nothing in the properties of the bases that determines their sequence by physical necessity. So I do think that sort of explanation really works.
Could you explain how do you explain these fossils? How do you explain these atavistic organs in the human body? Like, for example, the appendix, which are useful in lower animals, but now they are vestigial. How do you explain these in evolutionary?
Yeah, so the appendix is functional in the context of the human body. It's a reservoir for antibacteria. It's has a function in the immune system. So the appendix is not functional. Is not functionless.
Yeah.
So there are many organs non-functional, like the nictitating membrane in the eye, the appendages in the heart. They are non-functional. How do you explain, if the intelligent designer is there, why should you put these structures in the human body which are non-functional?
Well, I would be interested to hear an example. I think that, I mean, it's an argument from ignorance. We don't know what particular organ within the human body is doing, and therefore we just plug in Darwin as a placeholder for that of which you're ignorant. And then when we discover that actually there is a functional reason why it's that way, then the argument has to retreat with each advanced scientific discovery. So it's basically a Darwin of the gaps fallacy. But in fact, I think that I'm not sure what functionless organ you would have in mind when it comes to the human body.
Yeah, for example, in certain bones, there is a part in the back, the this shoulder blade bone, there's a part called atavistic. It's a very small part. This is functional in lower animals, but not in humans. And in the heart also, the appendage is non-functional in humans, but it is functional in lower animals. Why should an intelligent creator intelligent designer put such organs in a human body, which are
so that's what Jonathan is telling. Earlier, people thought this is all lot of vestigial organs, what Darwin said, that this is not functional. Now, the medical people and biologists have found lot of functions for that. So earlier, whichever was thought as vestigial and waste, like DNA, junk DNA, and many others, now it is functional. So we don't know whatever you're telling, there might be a function which is not known now. So might be research later might find. That's what I feel. So.
Exactly is the point. Whatever is not known, we should find out, not attribute to God. We should find out.
Yeah, that is right. But what I'm telling is, now we are not attributing that whatever is not there to God, but for every information, small information.
Will not bring an intelligent designer also there.
No, if
because
yeah,
we don't know the
right,
answer, we should not put an intelligent designer there.
No, we are not putting intelligent designer behind vestigial organs.
Maybe another question. If I can ask one more question, it will broaden this outlook. See, there are billions of galaxies. Why should an intelligent designer put human life only in the minuscule, the infinitesimally minuscule portion of the universe? Nowhere else. Why?
Okay, if you can give me 2 minutes, I can explain. See, what I wanted to tell is there are two choices. You can either think it is it's a choice, it's a chaos, or it is an intelligent design. There are two ways. So which one makes more sense when we see? So what we understand is, in all the galaxies still now, 5,000 exoplanets, planets rotating around their own sun has been found. But till now, why there are no life found? And why in the special planets so many specialities like greenhouse effect and magnetic field and atmosphere, everything, why it is at the right, why it is at the Goldilocks zone? There is some purpose behind it. So instead of attributing to chaos and chance, similarly, why in the DNA, if you see, it's much better than a language. So there is an information. So if a scientist in a proper way, as Jonathan is telling, if you research it, I think we tend to believe more towards the intelligent design than a chaotic design. That's what I would say.
It is just because it is chaotic, it is present only in this part of the world, universe.
So even if the rarity of life in our universe is evidence against theism, that evidence against theism is swamped by the positive evidence for theism, because it's far more probable on theism there would be life at all anywhere in the universe than it is on atheism. But in terms of the argument that you adduced against theism, namely the rarity of life in our universe, that doesn't seem to be a very compelling argument at all, because it assumes that God can only have one purpose for creating the universe, namely to populate it with life. And I think that a biblical concept of why God created such a vast universe is, as the psalmist says in Psalm 19:1, the heavens declare the glory of God, the skies proclaim the work of his hands. It's created for us to behold the glory of our creator.
Thank you so much for all the questions and the conversations that we've had this evening.